It is my distinct honor to introduce our second Dom Luke Childs lecture speaker. He is Dr. Jonathan, aka Jake Kurtis, MD, Ph.D., Portsmouth Abbey class of 1985. Dr. Kurtis is the director of the Center for International Health Research at Rhode Island Hospital and Brown University, and one of the country's leading researchers on the development for a vaccine for malaria. He is a professor of pathology and laboratory science with specialty training in transfusion medicine and coagulation at Brown Medical School. His talk today will focus on pediatric malaria. I am so happy that we have a real live scientist speaking to us today. Scientists are real people. Dr. Kurtis is a real person who is a world-renowned researcher and who is making a difference in the world by saving lives. Some of you might be thinking, hey, I want to make a difference in the world. I want to help people too. Perhaps Dr. Kurtis will inspire you to follow that dream. Two other things about Dr. Kurtis that are part of his claim to fame. First, Dr. Kurtis is the uncle of Emmeline. And second, Dr. Kurtis survived AP Bio while he was at the Abbey, and he lived to tell about it. Please join me in welcoming a fellow Raven, Dr. Kurtis.
Well, he said that I survived AP Bio. He did not tell you in what condition. So, I want to thank Kathy for inviting me back. You know, it's exactly 30 years since I graduated, which tells me that the statute of limitations on the transgressions which I committed while here must be up. So I'm thrilled to be back. And I'm going to talk to you about something that you may not know much about. Malaria. I assume everyone's heard of malaria, right? Yeah, pretty much. These lectures are typically fairly formal, right? I'd like to make this somewhat informal. I don't want to lose anybody when I present. Sometimes it's going to get a little bit technical. And so if I'm losing, just put up your hand and I'll stop, okay? We can make this a little bit more of a dialogue. This is a nice, comfortable audience. All right? So up at the Center for International Health Research, we work on vaccines for Falciparum malaria. It's the kind of malaria that kills. And it is overwhelmingly the most important single-agent killer of children on the planet. One child dies about every 15 seconds. It's stunning. It's the great culling machine of sub-Saharan Africa and Southeast Asia. So, let me see if I can figure out how this works. I didn't do the test right. Easy. So if you walk around the halls of a major university, particularly a medical university, you will see the bones of assistant professors who died academically while trying to study malaria. It's a very difficult parasite to study. It exists in difficult climates. I'll show you about that in a little bit. We don't have great animal models to study it. And for that reason, and the other reason is, if you look at how we fund science in this country, what do you think gets more funding? Research into male pattern baldness or malaria? There's another disease that afflicts older men that gets lots of research as well. As I get older, I'm thrilled about that. But see, we can keep this a little informal. Now that I've divulged all of those things to you, seriously for a second, we underfund malaria, even though it's the greatest single-agent killer of children on the planet. This slide, let's see if this works. Pointer. Yeah. This slide puts it into perspective. You guys have all grown up in the post-HIV era, correct? You know to be afraid of HIV, right? And you should be. Since the beginning of the HIV pandemic, approximately 30 million individuals have been infected with HIV. Since the beginning of the HIV pandemic, 50 million individuals have died of malaria. The HIV pandemic began in about 1984. How many HIV deaths were there in 1983? Close to 0. How many deaths were there from malaria in 1983? About 2 million. In 1973, how many deaths from HIV? 0. Malaria? 2 million. 1883, 1683, 1283. The same killing rate for malaria. Where are our research funds going right now? HIV. And that's good. My point is not to denigrate or diminish the impact of HIV, but rather to put it in a context of a disease that you're all familiar with, and rightly so frightened of. So the issue is not less research for HIV, the issue is more research for malaria. So, quickly, 74% of all childhood deaths occur in Africa and Southeast Asia. What do you guys think about that? Is that proportionate to the population distribution? It's really disproportionate, isn't it? Too much death in these particular locations. When I walk through the halls of Hasbro Children's Hospital up in Providence, you see children dying, and it's tragic. Are they dying of infectious diseases? No. They're dying of other things, typically cancers, traumas, not infectious diseases. Despite what you may read in the Providence Journal, flesh-eating bacteria are not yet overwhelming us. They might, but they're not yet overwhelming us. Children in the developed world do not die of infectious diseases. In the developing world, check this out. More than half of these deaths in the developing world are occurring from infectious diseases. It's the great elephant in the room that needs to get addressed. Hard to know how many children are actually dying from malaria, but it's something on the order of 2 million. It's sobering, isn't it? It's really sobering. Most important single agent killer, bar none. Nothing comes close. Not TB, not HIV, not even diarrheal illness or upper respiratory tract infections, which tend to be many different kinds of microbes cause those. So it really is the guy you want to take down. So a very famous health economist named Jeffrey Sachs coined the phrase, where malaria has prospered, people have not. When you think of sub-Saharan Africa, what is your vision? Poverty, misery, war, political instability. It turns out, this is a map of the malarious world. In green are areas where the parasite is resistant to our former good drug. So it's much harder to treat malaria. We can, but it's much harder to treat it. In mauve, or purple, are areas where the drug is still useful. The drug is called chloroquine. If you look, it's all in the tropics, and now I'm going to explain to you in a little bit why. It has to do with the transmission of the parasite. If you have an under 5 mortality rate of 25%, the places I work in Tanzania and Kenya, 1 out of every 4 children is dead by the age of 5, and they're dying from malaria. Just imagine the rent that tears in the social fabric of a community that suffered colonialism, post-colonial dictatorship, ongoing tribal conflict, and now 1/4 of your population is dead by the age of 5. Do you think that that society has a chance to actually develop into a safe and peaceful environment? It's a real problem. Is it a problem for us here in this room? I submit to you it is. Are you familiar with the recent wars that are occurring in sub-Saharan Africa that inevitably other nations get dragged into? It's not just a problem there. It's a global problem, and it has far-reaching influence. If you're going to talk about malaria, which is caused by a protozoan, which is a little tiny microorganism, you have to understand the life cycle. I torture medical students with this, but you have to understand a little bit about the life cycle. You get malaria from the bite of a female Anopheles mosquito. Gloria Steinem would not be thrilled with this. Only females transmit. Because, I don't know if you know this, only female mosquitoes bite you. Did you guys know that? They're all females. They are biting you because they need a blood meal in order to lay their eggs for oviposition. So that's what the blood meal is for. Here's the part that's really gross. Did you know that when a mosquito bites you, what's the first thing she does? She hacks a loogie. Why? Whoa! Head of the class, you're spot on. That's absolutely right. If she didn't anticoagulate, prevent the blood from clotting, her straw, it would clot, and that wouldn't work. So, the first thing she does is she spits down. It turns out there are scientists that are working on figuring out what the anticoagulants are in mosquito spit in order to anticoagulate typically older adults that need it, that are clotting too much. When she spits, this parasite is so slick. It's figured out that if the parasite's hanging out in the salivary glands of the mosquito, when she spits, in go the parasites. This form is called a sporozoite. Little thread-like things. I'll show you some pictures in a second. Those sporozoites do something remarkable. The mosquito bites you over here. Within 5 minutes, they've entered your bloodstream, passed all through your body, found the liver, you should know that, found the liver, and infected your liver cells. 5 minutes. It's remarkable. It's like you guys all getting to class on time. It's remarkable. So, 5 minutes. It takes 5 minutes. There's a vaccine that you may have heard about if you read the Wall Street Journal, for instance, or the New York Times. They talk a lot about a new vaccine called the RTSS vaccine. Has anyone ever heard of that? A couple of you guys have read about it? RTSS vaccine? It's nearing licensure in Europe. The RTSS vaccine is designed to make antibodies that block those sporozoites from infecting the liver cells. Can anyone tell me what a problem might be for the RTSS vaccine? 5 minutes. Those antibodies have to be ready to rock high concentration in order to interdict sporozoites from infecting hepatocytes. And that's been the great Achilles heel of that vaccine. And it's only a teeny bit not as good as a bed net protecting you from the bite of the mosquito. So it's not a particularly effective vaccine, but it's a nice try, and it's a good proof of concept. So sporozoites, they infect your liver. Over the span of between 5 and about 15 days, each sporozoite develops into 10 to 30,000 merozoites. Now the mosquito only spits 5 to 15 sporozoites in, so only 5 to 15 of your liver cells get infected. And over the next 2 weeks, like I said, they develop into these merozoite things, 10,000 to 30,000, that burst out of the liver cell, the hepatocyte. When I give this talk to the Brown University undergraduates, I make the comment that typically the Brown University, no one in this room, but the Brown University students typically do more damage to their liver on a Friday night than occurs, no one in this room, than occurs with malaria, because only 30 or 40 of your liver cells get burst open, and you've got trillions of liver cells. So, no disease yet. You're asymptomatic. When these merozoites pop out, each one wants to infect a red blood cell. Now remember, red blood cells are the cells in your blood that are delivering oxygen to the tissues, right? So each one of these wants to infect a red blood cell. There's a vaccine that you may have heard of that tries to block those merozoites, those little blue things, from infecting the red blood cell. That vaccine has a challenge. Do you know how long the merozoites are outside the red blood cell? 140 microseconds. Do you think that vaccine is going to challenge? The antibodies made by that vaccine have 140 microseconds to prevent that interaction. It's not going to work, I don't think. Still no disease. This is where the fun part comes. The merozoite infects the red blood cell, and it goes through a cycle. And for Falciparum malaria, Plasmodium falciparum, that's the malaria that kills, there are others that infect humans, but Falciparum is really the one to know about. For Falciparum, this cycle is a 48-hour cycle. And the merozoite enters, it forms what's called a ring form. It looks like a fancy high school or a college ring, you know, those signet cell rings? Ring form, then it turns into something called a trophozoite, and then into something called a schizont, and then it ruptures. 48 hours. What do you think happens when your red blood cells rupture? Your ability to deliver oxygen to your tissues plummets. And so if you have half of your red cells infected by parasites, 12 to 48 hours later, they're all going to burst. Your hemoglobin drops in half. It would be like bleeding out half of your blood volume. How do you think that would feel? Has anyone had malaria? It hurts. It definitely hurts. I'll talk a little bit about how it hurts in a minute. So this is the part where all the disease occurs. The vaccine I'm going to tell you about, I'm going to try and convince you we've identified a vaccine that doesn't block entry of the merozoite into the red cell, but rather blocks the rupture. It traps the parasites inside the red blood cell. It's like being trapped inside a burning house. If you're trapped inside the red blood cell and you can't get out, you can't multiply. Because remember, one merozoite goes in, about 20 to 24 merozoites pop out. Each one then goes on to infect another red blood cell. So you get this exponential wave of infection. And over the span of 72 hours, you can go from running a marathon to being dead. It's remarkably quick. So this asexual reproduction, this is all asexual reproduction, gets boring for the parasites, and every once in a while they get it on, and they differentiate. This is true, this is remarkable, I'm not lying. You can't make this stuff up. So they differentiate into male and female gametocytes, and only they can tell the difference. And what's really remarkable is the male and the female gametocytes, you'll look for hours on a blood film to find them. But along comes another female mosquito. She takes up a blood meal. These male and female gametocytes, they end up in the gut of this mosquito, which to them is like candlelit dinner with Liberace on the piano. It's so romantic that they decide to conjoin, forming an ookinete in the belly of the beast. Over the span of about 2 weeks, that egg, if you will, differentiates into the form called the sporozoite that migrate up to the salivary glands, ready to repeat the cycle. There's a vaccine that's designed to attack the form in the mosquito. So you make the antibodies to the mosquito form. They don't do anything to you. They get taken up in the blood meal. Inside the belly of the mosquito, if the ookinetes form, the antibodies attack. So it blocks transmission. It's an altruistic vaccine. Does that make sense? It's crazy, isn't it? That one might actually do something. That one seems to be working. But remember, it won't protect you from disease. It'll protect you from transmitting to your friends. Okay, so that's life cycle. Any questions about that? All right, let's talk about how you make people sick. Well, first, a few pictures. This is a female Anopheles mosquito. That's her proboscis, her drinking straw. That's what she has to anticoagulate. You can see the male's one is totally wimpy. He's never going to suck blood through that. But it's always fun to make fun of the men. The parasite is sitting in the salivary gland of her head, ready to go down. This is what the sporozoites look like. They're thread-like forms. They're motile. They can swim around. And like I said, it's amazing. Within 5 minutes, they're out of your circulation into your liver cell. This is what a blood film looks like. These are red blood cells. These are, that's a ring form. Does it look like a ring to you guys? These are parasites. And if you notice, half of this person's red blood cells are infected. What's going to happen in 48 hours? Half of their hemoglobin will be gone. You guys and gals, healthy youngsters, we could bleed you out half of your blood volume. You wouldn't be fine, but you would absolutely survive, and you might even be able to go to class. Where's the school nurse? But do that to someone who's sick already has a low hemoglobin, hasn't had a meal with iron in it in 6 weeks, and you understand that this can be fatal very quickly. It took me forever to find a particularly sinister-looking parasite, but I did. I think they're all sinister, actually. So this is a true story. Any of you guys thinking one day you want to become a physician? Who wants to become a hedge fund manager? How many physicians do I see? None. Oh my God, we got to work. 1, 2. Don't be ashamed. 3, 4. Good. All right. If you want to become a physician, pay attention for a second. This is a true story. My dad's a pathologist. That means they generally study the dead people. This blood film came from a guy who presented at his hospital, told the ER doc, I've been in Kenya. I had malaria in Kenya. I feel like I have malaria again. but he had watery diarrhea, which is not a classic symptom of malaria. He was sent home without checking for malaria. He came back later. If anyone wants to hear the full details, come up to me afterwards. This was his blood flow. Do you think he survived? He did not. The physician was told the diagnosis, and he ignored it. So, physician hubris, beware. It gets out of control very quickly. You can look fine, and 48 hours later not be so fine. This is what the gametocytes, this is the sexual forms, look like. And so a mosquito has to take up 2 of these in order to transmit. All right, so mild malaria is like the worst flu you've ever had. Myalgias, that's muscle pains, bone pains, fevers that can be recurrent or not, that's a bit complicated, and severe headaches. That's mild malaria. How does it kill you? It causes syndromes, things like cerebral malaria, severe anemia, placental malaria, algid malaria, pulmonary malaria. Notice it's all falciparum. The other 2 species are vivax malaria, and there's a fourth called ovale. But almost all the killing is falciparum, Plasmodium falciparum. This is how you die, syndromic. Why is it syndromic? Well, when the parasite is inside a red blood cell, it wants to avoid your spleen. So if your liver's over here, your spleen's over here. The spleen is the area that picks off infectious agents and gobbles them up. And so if you're a parasite inside a red blood cell, you want to avoid the spleen, right? So one way to avoid the spleen is to stick everywhere else. And so what this parasite does is it puts up proteins on the surface of that red blood cell that are sticky to your capillaries. You guys know what capillaries are? Little vessels that move blood around. So your parasites stick in the periphery, avoiding the spleen. It gives them a huge selective advantage. Here's what it looks like. So this is a red blood cell with a scanning electron microscope picture. That's a healthy red blood cell, that's a healthy red blood cell. This one has parasites ripping out of it. See the hole? That's a parasite coming out. These things here are the parasite proteins. They're called knobs. Those knobs allow this infected red cell to stick in the periphery. Now imagine all my red cells are sticking in the vessels that give blood to my finger. What do you think will happen to my finger? It'll turn blue, right? It'll die. It'll eventually turn black. So that would kind of sting. But this would be worse. Another clue, by the way. If you're ever shown a photomicrograph of a brain section, this is brain. It generally means bad outcome. Generally means bad outcome. This is brain. This is sort of normal looking brain tissue. See that hole there, that big vacuole? That's dead brain. This is a blood vessel. All these blue things with dots in them are infected red blood cells that are sticking to the capillary wall. Do you think any good blood can get through there? No, you need Drano. That is a mini stroke occurring. Has anyone had cerebral, or this is called cerebral malaria? Has anyone had So cerebral malaria has a case fatality rate of about 40%. And in about 10%, and I can personally attest to this, I've had this disease, in about 10% it leaves you with profound neuropsychiatric illnesses. I can personally attest to that. Yeah. You guys have to wait, right? Dead brain. All right, this is fun. So I use this one to teach the medical students. And we had a medical student in our group who studied malaria with us, and then he was taking his med school classes. So I figured Dave would know the answer to this. I always ask, what's the tissue? And none of the medical students ever know what tissue this is. So then I tell them, I give them a hint. I said, well, and this is actually, in my day it wouldn't be true, but today it is true. Only about half of you in this room could ever have this organ. And my medical student, Dave, raised his hand. And I said, Dave. And he goes, kidney. If you want to know his last name so you can avoid him as your physician, I will be happy to. This is a placenta. This is a placenta. This is the baby inside. So that's the trophoblast layer. That's baby blood, mommy's blood. That's the barrier. You talk about the placental barrier, for the mommy-baby barrier. That's it. You're looking at it. It's skinny and thin. All the good stuff has to go from mom to the kid. All the waste has to go from the kid to the mom. And apparently I'm told that proceeds right on through puberty. My oldest is 12, so I'm hoping that we have a slightly different age plan there. Do you guys see all these red dots? Well, first of all, look down here. See all these red dots? Those are red blood cells. Perfectly healthy inside the baby. Do you see the red dots up here? What's inside that? Those are parasites. There are parasites that, in fact, stick to the surface of that placental cell. And if that thing gets all stuck, no good material goes to the baby, and none of the waste comes to mom. It's a leading cause of maternal mortality. Women typically don't die of childbirth in this country, do that? It's common in the developing world. Common. Big cause of maternal mortality, huge cause of infant mortality is having your placenta locked up. Okay, so this has been gloom and doom, right? Why is it not uniformly fatal? It turns out children, at some point in their development, make antibodies which first block disease, so they still have the parasites but they're not as sick, and then block infection. And it's mostly antibodies. Cellular immune cells might have some component, but the real question is, if antibodies can protect kids, can we make young kids, if antibodies can protect old kids, can we make young kids look like old kids immunologically? Can I make a young kid make a protective antibody? That's the extant question. So, you guys should all know that vaccines are the most important single health intervention on the planet, bar none. Even the WHO buys into them. They've had enormous impact in getting rid of diseases like meningitis, polio, measles, although in our country we still have measles outbreaks, which is a shocking indictment of our healthcare system here. Amazingly effective. They don't require continued infrastructure support, and if you're working in sub-Saharan Africa, that's huge. But do we have a vaccine for any parasitic human infections? No. Any idea why? The research money doesn't go to parasites. It goes to pattern baldness. So, we might want to rethink that. Especially background. You guys are familiar that scientists often use animal models to study things, right? I'm here to tell you that mice lie. We have had an outstanding vaccine for malaria for now 4 decades, 40 years. No rodent need live in fear. It has not translated or generalized to humans at all. It's been 40 years down the tubes. So we can protect mice. That doesn't necessarily mean we can protect humans. If you take, and this was done by the Army, it was run by a colonel and all these study subjects were privates. I am sure there was no coercion, however. If you take infected mosquitoes and you irradiate them so that the parasites inside are wounded, and then you stick your hand in that bag of mosquitoes, and so you have 1,000 bites. You'll pull your arm out and it'll look like Arnold, right? It'll be all inflamed. 1,000 bites. Those sporozoites will go to your liver and die. If you do that, and then challenge yourself with live, healthy sporozoites, you won't get malaria. So we know that we can prevent this disease. That's called an attenuated vaccine. But can you imagine trying to vaccinate 300 million individuals in sub-Saharan Africa with irradiated mosquitoes? It's just not possible. It's not tractable. So we need to figure out what are protective immune responses. So in science, if you're going to make advances, you first have to stand on the shoulder of giants, paraphrasing Newton. But also paraphrasing Newton, you have to believe that you can see further. So it's this weird ego trip. You've got to think that the paradigm you're working in isn't sufficient. And it's pretty clear the malaria vaccine paradigm, as it currently stands, is not sufficient. It needs new ideas. And so we tried to come up with some new ideas. We, like Mephistopheles, are fans of man, as opposed to mankind, not rodents. And so we go into endemic villages in Kenya and Tanzania. We go into an endemic village where 50% of the kids will have malaria at any given time if I just take blood films. And we look. Some kids get lots of malaria. Some kids don't get lots of malaria. And we try and figure out what is it that's different between the kids that are getting lots of malaria and the kids that are getting no malaria. Some of it is, well, the kids that get lots of malaria probably live where the mosquitoes are, right? By the water, by the lakes. That's a big component. However, there are other issues. It may be that some kids, let's say this half of the room, doesn't get a lot of, well, that's me. This half of the room doesn't get a lot of malaria. You guys have antibodies that protect you. Sorry guys, you don't. Who's clapping for that? So the idea is, can I figure out what those antibodies are? And since they've been so nice and attentive, can I vaccinate them so that they make these antibodies? Can I make them look more like you? I'm noticing a huge gender divide down the hall. So that's the idea. So we identify who's resistant, who's susceptible. We figure out what parasite genes encoding parasite proteins are being recognized by the antibodies in the resistant people that the susceptible people don't see. And then using what I call molecular DNA gymnastics, the details of which are not that important. We take that parasite gene, we make tons of that parasite protein, and I immunize these guys. If I immunize you with the parasite protein, you should make that antibody to the parasite protein, right? And if you do, you're going to behave like them. You'll be resistant. That's the whole idea. That's the whole idea. You guys get that? That's the whole idea. All right, so here's what we did. We enrolled, how much time do I have? All right, we can go. We enrolled a birth cohort of almost 1,000 mothers as they deliver in Muheza, Tanzania. So just to orient you, that's the Indian Ocean, Lake Victoria, Tanzania, Kenya, Dar es Salaam is here. That's Muheza. It is truly a place that was at least not paid enough attention to by the good Lord. It's really forsaken. The entomologic inoculation rate, that means the number of infectious bites per person, is 400. 400 infectious bites per person per year. That's more than one infectious bite per day. Think of the great thumb of malaria pressing down on this poor population. The under 5 mortality rate in this community is 25%. It's crazy, isn't it? It's unsustainable. We enrolled almost 1,000 when we were finished, moms, as they're giving birth. And we followed the children from day of life 0 every 2 weeks with a blood film. We detect parasites on the blood film, right? Every 2 weeks we collected blood films from these kids in the first year of life, and then every month in years 2, 3, and 4. So it's a 5-year study. These kids were giving birth in a regional and district hospitals. They're spread out over a 50-kilometer square area, huge catchment area. We provided active surveillance for their health. And what I mean by that is, since they're spread out, I'm talking mud huts, straw roofs, dirt tracks that are struggling for 4x4 cars to get to, Jeeps. So it's very remote. No electricity, no running water. No health care to speak of. We had a health wagon, like an ice cream truck, except it was filled with health stuff. We had a health wagon visit every kid every 3 days. So we had 6 wagons moving around this 50 square kilometer, and the wagons were staffed with drugs that weren't counterfeit. Unfortunately, a lot of the drugs in the developing world are counterfeit drugs, so they don't work. And it was staffed with a clinical officer, kind of like a nurse practitioner, someone you might see when you go to the pediatrician as a first pass. Despite what we thought was outstanding health interventions, this is the survival curve. This happens to be guys that were iron deficient and iron replete. It was better to be iron deficient because the parasite needs iron. This is survival by weeks. And what you see, it's hard to read it, but 10% of our cohort died in 4 years. Under the best possible health intervention circumstances, I felt so good about being able to provide that. However, was it enough? No, it still sucked. 10% of my kids are dead. It's like an overwhelming. Or you guys you should be blown away by how horrific this is. Under the best possible circumstance that I could imagine. So, we tried our best. We still had huge mortality. Okay. This slide shows you parasitemia in the vertical axis and age of the cohort. Young kids have low parasites. 6 to 12 months old have more parasites. It peaks at the 1 to 2 year age and then falls off. Why do you think the young kids are protected? Any ideas? Yes? Mom's antibody. That is the prevailing hypothesis. No one's actually proven it yet, shockingly. But that overwhelmingly must be the right reason. So mom passes antibody. Remember, mom's protected. Mom passes antibody to the kid's protected. The antibody wears off after about 3 to 6 months. So that's why the kids we think are protected. And then, the young kids, then they start getting lots of infection. Notice something happens at the age of 2, and the kids are not protecting themselves. So we said to ourselves, let's look at these 2-year-olds. Let's pick the most resistant and least resistant based on the blood films that happened afterwards and collect 2 groups, and we did that. These are 2 groups, a group of resistant kids and a group of susceptible kids. In the interest of time, I'm going to take your attention to the bottom. This is the parasite density in resistant kids between the age of 2 and 3.5 compared to susceptible kids. So 12 kids, no parasites, 11 kids, lots of parasites. And I took their serum. That's where your antibodies are. And I had serum from resistant kids here, pooled, and serum from susceptible kids here. And the next thing I did, I actually had my MD Ph.D. student did this, the poor guy. I'll tell you why in a second. So he took all the parasite genes and he spread them out in a room, not unlike this room. Look down on the floor for a second. See all the squares? Each square has a parasite gene on it. Then he takes a filter and he throws the filter over the whole floor. The filter tricks the gene to get turned into a protein. So now every square has a parasite protein on a piece of filter paper. He pulls that filter paper up and he lays another piece of filter paper down. Pulls that filter paper up. He has 2 copies of the filter paper. One he probes with the resistant serum. One he probes with the susceptible serum. He's looking for parasite proteins that are uniquely recognized by the resistant serum, resistant plasma here. The red dots. So these are identical copies. The dots are just squares on a floor. The red dots are parasite proteins that only the resistant people see. This half of the room sees the resistant serum. It sees the resistant clones. This half doesn't. Does that make sense? It's called a differential screen. What I just described is a whole proteome differential screen. I want to know what these guys see that you guys don't see. And so we did that. My PhD student screened 1.25 million parasite genes. Imagine 1.25 million of these squares. He screened 1.25 million of them. And he found 3 parasite genes that only this side of the room, the resistant side, saw. 3 out of a million. Most of the genes were seen by you guys as well. If both sides see a gene, is it an interesting gene? No, it can't be because you're susceptible, remember? All right. So we did that. We found 1 gene. Here's a picture of what it looks like graphically. We saw that little piece. We call it SEA, Schizon egress antigen. Once I had that piece of gene, using molecular biology tricks, I can make the protein. And I can make it in vats, huge quantities, like a pressure cookers. I make huge quantities of that, what I think is a protective parasite protein. I think if I inject that parasite protein into this half of the room, they'll be protected. But before I do that, I have to prove that it's in fact a parasite gene. When we got this gene, you can query a place called GenBank that has all the sequences of everything that's ever been sequenced. This gene shows up, but no one's ever shown that it was a parasite protein. So the first thing I did is I took antibodies to this protein. It's called SEA, Schizon egress antigen. I took antibodies to this protein, and I probed, in this lane, infected red blood cells. In this lane, infected red blood cells. In this lane, uninfected and uninfected. Antibodies to SEA, irrelevant antibody. SEA antibodies see a single protein in infected red blood cells that they do not see in uninfected red blood cells. What I'm proving to you here is that this gene does in fact code for a parasite protein. So it's real. So I did that. The next thing we do is we take antibodies to this gene and we take parasites in culture. We grow parasites up in Providence. If anyone ever wants to see some parasites, come on up. Grow them in culture. You actually grow them in human blood. So you take the human blood infected with the parasite, you add antibody, and you come back later and you see how the parasite grew. So in this vertical axis, in the vertical axis here is number of parasites. Here is if I treat it with nothing. This is if I treat it with anti-sera taken from the animal before I immunized it with our protein. And here's if I take anti-sera from the animal after I immunize it with the protein. So this is specific antibodies to my gene. This has no antibodies to my gene. You guys see a difference in growth? Which group would you guys like to be in? This group or this group? This group. Half the parasites aren't growing. So to make my analogy back to the room, the folks over on this side look like this. They have the antibody. The folks on this side look like this. They don't have the antibody. The growth rate of the parasite is interdicted by about 60 to 70%. I'm running out of time, aren't I? All right. Remember I told you that there are different life cycle stages? There are rings, there are merozoites, rings, trophozoites, and schizons. We took antibodies to SEA, and we probed these different stages of the parasite. I thought that we were blocking infection. I thought we were blocking the merozoite from getting into a red blood cell. And so, if that's true, if you're blocking infection, what stage would you expect to light up? What stage does the infecting? You got it? Merozoite. I expected the merozoite to light up. I expected the merozoite to light up. This is the lane with my anti-sera. So you're looking, I expected green up here. And my poor postdoc, his name is Deepak. He brings me this data and I say, oh, Deepak, this is all wrong. You gotta go do it again. So he does it again. Same results. I said, Deepak, this is all wrong. You gotta do it again. He reminded me of Einstein's quote. You know, the definition of insanity is repeating the same action over and over, expecting a different result. So he actually said, then he said, no, Dr. Jake, you must come to terms with the data. So, after walking outside and stomping my feet, I decided to come to terms with the data. It's binding to the schizon. How could you stop growth if you're binding to a schizon? And that's where we got the idea maybe it's trapping the parasite inside the red blood cell. Sorry, I'll skip by some scanning EM. So this is another growth assay. This time you start with the trophozoites. In 12 hours, all those trophozoites would grow into rings. They'll pass through the schizon stage. We add antibodies to the trophozoites, and we come back in 12 hours, and we count schizons. If you add no anti-sera, all the parasites should have no longer be schizons. They should be rings now, and you don't count them in the bar. They should all have passed through the schizon stage. The pre-immune sera, they all pass through the schizon stage. With the anti-sera against SEA, they're trapped as schizons. They get trapped as schizons. Here's a picture. This is what pre-immune sera would look like. See those rings? Those are rings. The post-immune sera, those are schizons. And they actually look like they're dying schizons. So our antibody traps the parasite as schizons. I want to share 2 more slides with you. I'll skip over the mouse work. We went back to the 785 or so kids. And we measured antibodies to SEA, our protein, in all 785 of them. And we asked the question, if you had antibodies naturally occurring to SEA, how much severe malaria did you get? This is actually interesting because I had to fight with the journal about publishing this for an issue that you guys are probably very familiar with. So if you had undetectable antibodies, that means no antibodies to SEA, you had lots and lots of severe malaria. Severe malaria is literally 1 step from falling off the cliff. You're almost dead. Has a case fatality rate of somewhere on the order of 60%. So if you did not have antibodies, lots of severe malaria. If you did have antibodies to SEA, you see that line there? I had to fake it. The actual number was 0. But I had to put a little bit of a bar so you could see that there was data there. 0. There were 0 cases of severe malaria. The statistician at the journal wanted what's called an odds ratio, where it's the number of cases in one group over the number of cases in the other group. And so I had to write back to them that Mrs. Lupner in 3rd grade taught me that thou shalt not divide by 0. How do you do it if there were no cases? So it was a bit of a fight with the statistician over no cases. So no cases if you had the antibody of severe malaria, no cases of death, and no cases of malarial attributable death. Almost all the death is malarial attributable. So what do you think of that? Which group do you want to be in? The group with the antibody or the group without the antibody? None of you guys died, and lots of you guys died. So it's much better to be in that group. So I want to leave you with the idea that if we can vaccinate folks who don't have antibodies to this protein, that they might become protected. I want to leave you with some sobering thoughts. One is, Falciparum malaria is the great killing machine of sub-Saharan Africa and Southeast Asia. There are no magic bullets on the horizon. In fact, something very scary is recently occurring. There are parasites in Southeast Asia that are moving to Africa that are resistant to all known drugs. And so that really will be a major catastrophe for malarial morbidity. Multidisciplinary approaches. I've only told you about a scientific approach. The health economists have a key role. The demographers, the social scientists. You know, one of the best things we can do right now is distribute bed nets that protect people from getting bitten by the mosquitoes when they sleep at night. But it turns out, if you give bed nets to fishing communities, guess what they use the nets for? So we need social scientists to help us with those kinds of problems. Delivering bed nets alone, in theory, sounds simple, but in practice is quite complicated. So my point to you is that no matter what area of activity you're interested in, whether it be economics, policy, social sciences, epidemiology, all of them, as well as the basic sciences, can be brought to bear for this particular problem. Despite the recent awarding of a Nobel Prize for the discovery of a new anti-malarial, you guys familiar with that this year? So the 3 prizes for medicine this year, 2 went to the folks who discovered a drug for river blindness, and the 3rd was a Chinese lady who in the 1970s, in basically a concrete hovel of a research lab, discovered by reading an ancient Chinese pharmacopoeia from 2,000 years ago, the extract of a bark that is now our latest anti-malarial drug. So Nobel Prizes are available if you seek to pursue them. Sadly, despite these little bits of good news, malaria will continue to be the great killing machine for the foreseeable future until we come up with some better approaches. I'd just like to thank all the folks that were involved. First of all, I want to shout out to Bob Soms. I got interested in biology and medicine. Yes, I know, it's creepy, but it's true. Here, as a high school student, I got really turned on to the possibility of new discovery, and so I really want to thank Mr. Soms for that. I've been fortunate to have outstanding mentors over the years. Patrick Duffy and Mikhail Fried were my postdoctoral mentors and continue to work with me on this project. This is Deepak, the guy who told me to come to terms with the data. We gotta get his picture down, I think. And Chris Nixon was a PhD student. A pile of undergraduates helped out in all the, I didn't even show you most of the data, all the cloning and such. Pretty pictures from some folks up at Rowan Hospital. And the American taxpayer for some of the research support that went into funding the study. And with that, I will take any questions you might have. That's from 25 years ago. That's Pat Duffy in the middle of that. Questions? Please.
And the last part, can you elaborate more on how science is funding your work?
Yeah, that's a real challenge. So what is supposed to happen is there's some rank prioritization of diseases. And the problem is, if you have, let me think of a rare tumor, you've got a chordoma. 1 in 800,000 people develop that. But to you, it's extremely important. How do you rank order them? One way is this notion of something called a DALY. Has anyone ever heard of a DALY? A disability adjusted life year. It's kind of a macabre approach because you have to take each disease and figure out how much misery does that cause over the lifespan that you would expect to live for that particular disease. So things like breast cancer shoot to the top because it can occur in young women and it can cause morbidity for huge lengths of time. Things like prostate cancer go to the bottom because it typically occurs in much older individuals and it doesn't necessarily kill you. In fact, it typically doesn't. So that's one way to rank order. The problem is infectious diseases in particular are underfunded by industry. So do you want to make a new Lipitor drug, which is a cholesterol drug that you will take from the age of 44 till you die at age 80, every day? Or do you want to develop a new antibiotic that you'll take for 10 days if you're doing it right, once? So it's this area of economics that doesn't function for the group when left alone. It requires some kind of control. Funding in terms of NIH approach, NIH does attempt to look at diseases elsewhere on the globe, particularly things that might end up in a senator's son. So that is why HIV, or senator's daughter, That is why HIV rocketed up in terms of funding, frankly. I don't know if that quite answered your question, but there really is no good answer, to be honest with you. It's a lot of lobbying, too. Please.
Can you take us back to the beginning of your career? You're coming out of med school. What made you pick malaria instead of male pattern?
So, well, I looked in the mirror and I realized that one wasn't going to be my disease. I had the good fortune. I was on a night train going from Nairobi to Mombasa. And this is back in before you had all this nonsense. I hadn't spoken to an American in about 3 months. I was living in Kenya doing a coral reef ecology project, actually. First bit of science I'd ever done. Learning Swahili, politics. So I'm on this night train and I met up with some Americans. I hadn't seen them in so long. I wanted to hear news of home. And so we stayed up in the dining car late, and the dining car served things other than food. And so I went back to my cabin, the sleeping car, it's a night train, and I had such a bad headache. And I thought that, God was visiting upon me just retribution. But I get into the station. I mean, I'm actually answering your question. I get into the station, and I'm 18 or 19 years old, allegedly a strapping young man. I couldn't lift my rucksack, so I hired a porter to lift my rucksack, which is weird, right? I had rented a mud hut on the beach where I was doing my coral reef ecology work. I get to the mud hut, and then I don't remember anything for the next 3 days. I had cerebral malaria. I was in a coma. And it turns out that my travel physician up who ended up becoming my Ph.D. advisor, had told me, if you have a fever that's unexplained, take these 3 pills. They were curative medicine for malaria. So I must have listened to him, because I took those 3 pills. 3 days later, I wake up, I can barely crawl across the room. And there's no phones, there's no help. And so it took me about a week before I was really able to get back on my feet. And so what I realized was, number one, I love science, because I was actually doing a coral reef ecology project, loved it. And I looked around and I realized that this is the greatest problem of, in my opinion, humanity. If you're losing your 2-year-old children, how are you possibly going to have an effective society? And so for me, it was like a lightning bolt, galvanizing my interest in science and tropical infectious diseases all at the same time. Once I came to that conclusion, the rest of it sort of, then it becomes an engineering problem, not a physics problem. And it's just how do you get to the position you want to be in, which is ultimately to work on a disease like that. So I look at cerebral malaria as being the greatest thing that ever happened, except for the psychiatric. Hello. Please.
Going back to the thing you were talking about, the guy who had malaria before and then when he had it again, how common is that for your kids to come back?
Oh, that happens all the time. He probably had a new case. So this happened to me on another occasion. I actually developed malaria flying back, which sucks on a plane. Like, plane rides stink to begin with. But then to get malaria on a plane, the worst part of it was I was sitting next to a German anesthesiologist who had no compassion whatsoever. None. I'm like laying on the guy. He's like, jerk. So getting back to your, he probably had a new case of malaria. On the plane, goes to the hospital. He's got watery diarrhea. He's diagnosed with traveler's diarrhea. He's given Pepto-Bismol, which is the right thing to do if it's traveler's diarrhea. Sent home. He's an artist. He keeps a journal. Remember, did I tell you he was making films in Kenya? He's an artist. He keeps a journal. He writes in the journal. I don't think they quite got it right. 48 hours later, does that number ring a bell for you in terms of that cycle? 48 hours later, he goes back to that ER. Same ER. I'll tell you his name, but he's no longer practicing medicine. Back to that ER. Bloody diarrhea. He's diagnosed with invasive E. coli. That happens. Anyone ever have dysentery? Occupational hazard of working in the tropics. I'm an intestinal cripple. He's treated with an antibiotic, which is the right thing to do, the right kind of antibiotic for the wrong disease. What do you think was really happening to his gut? Remember the plugs, the hole, the vessels getting clogged? He had ischemic bowel. His bowel was dying and sloughing off. He goes home, keeps the journal, writes, I think they're really messing it up. Comes back. That's unfortunately what my dad saw. Finally, a different ER doc decides to do a 3-cent test, which is a blood film. And you look at it and you go, oh my God. So that was the story behind it. Please.
And malaria has tracked over hundreds of years and killed more millions of
Yeah, I'm going to repeat it. So the question, if I understood it, was, so if there's this great malarial protective protein, why the heck is the parasite still expressing it? It should have been deleted by natural selection. You shouldn't show your soft underbelly to the immune system. I wasn't sucking it in when I did that. My timing was all off. Is that your question? All right, so I'm going to answer it 2 ways. 1, what proportion of the very young kids made antibodies to it? No one asked me. 6%. So it's not immunogenic under natural conditions. Number 1. Number 2, imagine I'm attacking your finger. You might just cut off your finger, right? Now imagine I'm attacking your heart. So the experiments I didn't show you is we genetically removed this gene from the parasites. They can't grow. If I decrease the expression of this gene in the parasites, they're crippled, they barely grow. So this gene is absolutely essential for the parasite to escape from the red blood cell. So it can't avoid making this protein. It is a local maxima of natural selection. It can't get off the hill.